This section explains how a silent process becomes a countable one, turning the invisible accumulation of somatic mutations in an aging animal into a number you can read off a plate. Somatic mutations accumulate invisibly across an aging tissue. No microscope counts them. The chapter's move makes them countable. It carries a functionless LAC-Z gene on an excisable plasmid seeded throughout the mouse or fly genome. You let the animal age, cut the plasmids back out, and shuttle them into Escherichia coli, where each mutated copy flips a single bacterial colony or each corable phenotype. Mutation load becomes a colony count. The reporter's uselessness is the whole point. Because LAC-Z does nothing in its host, selection neither culls nor spares its mutants, so the count reads the genome's raw, unbiased mutation rate. Any invisible rate yields to this pattern. Bind it to a neutral passenger, then read out where one lesion equals one binary flip. This section explains how a mutation, invisible on its own, is turned into something you can count. A mutation stays invisible until something makes it countable. The trick is to sew a bacterial LAC-Z gene into the animal's own chromosomes on a record out to a substrate that can score it and accumulate damage. Then excise the reporter back into the bacterium, E. coli, where positive selection turns each mutant into one colony you can tally. The organism becomes a passive mutation recorder. The plasmid is the shuttle carrying that record out to a substrate that can score it. The generative move is this: read in vivo, count ex vivo. It cracks open tissues that never divide, the liver, the heart, the brain, which dividing cell assays could never read. And the payoff is a surprise. Near half of the mutations that aging tissue accumulates are large genome rearrangements. Not point changes are large genome rearrangements, not point changes. This part explains subtractive hybridization as a way to find senescence-specific genes. The core idea is a set difference computed by chemistry instead of enumeration, to find which genes a senescent cell expresses that a young one does not. You do not read both catalogs and compare. You let the transcripts cancel. Excess messenger, RNA from the young population, called the driver, is hybridized against the senescent one, called the tester. Every sequence common to both finds its complement and pairs off, dropping out of solution. What survives unhybridized is exactly the residue, genes on in senescence, silent in youth. Annealing kinetics perform the subtraction, and abundance sorts itself, since the commonest transcripts saturate their partners fastest. Swap driver and tester, and the same machinery hands you the youth-specific genes. The method is symmetric in what it deletes. This section explains how you read a senescence program by taking the set difference between two populations of transcripts. Senescence is a program of differential expression. To read it, you want the set difference between two transcript populations, the genes switched on in the senescing cell, which we call the tester, and silent in the reference, which we call the driver. Suppression subtractive. Hybridization computes that difference, tester minus driver, physically. You mix the tester with an excess of driver, denature, everything, and let complementary strands re-anneal. Shared sequences pair off with the driver and drop out, so only the tester, unique cDNA, survives to amplify. The subtraction is base pairing itself, to amplify. The clever part is the normalizer riding along. Re-annealing obeys second-order kinetics, so abundant transcripts pair fastest, which flattens the abundance spread. A rare gene ends up as visible as a loud one. Difference plus rate equalization, run in a test tube. This section explains how genetic variation in Drosophila lifespan is traced from a broad statistical signal down to a single nucleotide. A lab mutation proving that a gene can set lifespan says nothing about whether that gene does vary lifespan in the wild. Induced knockouts and naturally segregating variation are different populations of cause. This chapter is the descent between them, a resolution ratchet running from statistics down to the nucleotide. The descent proceeds in 3 steps. Recombination. Mapping localizes the variation to a chromosomal region holding tens to hundreds of genetic labor. Quantitative complementation testing collapses that region to a single gene. Linkage, disequilibrium mapping, collapses the gene to the causal nucleotide, the QTN. Each rung trades breadth for resolution, buying causal precision with genetic labor. The DDC anchor walks the whole descent from QTL to positional candidate to natural polymorphism. Its place in the book is to turn the question of which pathways can matter into the question of which alleles actually differ. This part explains quantitative complementation, a way to localize a genetic cause inside a region too coarse to name a single gene, working by subtraction rather than by scanning. A longevity, QTL, from recombination mapping still spans tens to hundreds of genes, so you cannot read the causal allele straight off the map. The move is this: cross both parental strains against a deletion that covers part of the region and ask whether the 2 strains' lifespan gap changes when one copy is removed. That is a strain by deletion interaction in a 2-way analysis of variance. If the gap changes, the causative allele sits under that deletion. If it does not change, there is no QTL there. Then iterate the deletions inward and afterward swap in null mutants of named candidates, collapsing region to gene to nucleotide. Failure to complement, read as an interaction, is the probe. This section explains how the genetics of a continuous aging trait get pinned to specific places on the chromosome. Variation in a continuous trait, here, the rate of thymic involution, the shrinking thymus that serves as the cleanest universal biomarker of immune aging, is heritable. Its causes are localized by asking which chromosomal regions co-segregate with the trait across strains. That is the whole move. A quantitative trait locus is a genomic address whose alleles shift the mean of a distribution found by correlating genotype to phenotype. The generative trick for aging is the recombinant inbred panel, the BXD mice. Genetically identical, replicates let you sacrifice the same genome at successive ages, so a longitudinal decline becomes a repeatable number you can map. Aging. Phenotype in, chromosomal region out. Mendel's discrete logic stretched onto a continuum. This section explains how a genetic map recovers the causes of an age-related trait. The whole move is one trick: freeze the genome, vary the age. A quantitative trait like thymic involution has no single gene. Its variance smears across many loci. QTL mapping recovers them by correlating the trait's variance against the variance of genetic markers across a segregating population. Where a marker's strain distribution pattern tracks the phenotype, a causal locus sits nearby. The chapter's leverage is the BXD recombinant inbred panel. Each strain is a permanent, genetically identical clone line, so you can sacrifice and remeasure the same genotype at successive ages and fit involution to a negative exponential curve. This is a destructive longitudinal assay made repeatable by genetic replication. That replication is precisely what buys the mapping resolution. This section explains how two-dimensional gel proteomics turns an unknown proteome into a readable map. Two orthogonal separations do the work. Isoelectric focusing sorts proteins by charge. Then SDS, Page sorts them perpendicular to that by mass. A spot becomes its own address given by its isoelectric point and its mass. Aging signature becomes geometric. A protein that has grown more abundant darkens its spot. One that gains an oxidation or splices to a new isoform migrates to a fresh coordinate. Mass spectrometry then reads identity off whatever moved. The generative move is discovery without a candidate. You never name the protein first. You let displacement nominate it. The grapeseed extract rat brain study anchors this. The proteins. The supplement targeted surfaced with no a priori guess about which ones would. This part explains how two-dimensional gel electrophoresis turns a protein mixture into a searchable address space. Spread 1,000 proteins across a gel by one property, then across the perpendicular axis by a second independent one. Charge, the isoelectric point, comes first, then mass. Each protein falls to a unique coordinate that no single dimension could resolve. Two orthogonal separations factor the mixture, and the spot map becomes an address. Factor the mixture, and the spot map becomes an address space. The payoff is discovery without a hypothesis. Compare young against old gels, and the spots that shift in intensity or multiply into isoform trains name themselves. You never had to guess which protein to assay. This is the same move as any orthogonal basis decomposition. Independence of the two axes is what buys unique addressing. The grapeseed extract study anchors it, with protein targets surfacing under no a priori candidate. This section explains how metabolomics changes the order of operations in aging research. Aging research classifies by fiat. The experimenter declares an animal old or young, long-lived or short-lived, fed or restricted, then studies the difference. But every such label is imposed a priori, using external, often self-referential criteria, chronological age, or the diet you happen to serve, criteria that do not transport across strains or to humans. Metabolomics inverts the order of operations. Instead of bringing a hypothesis and measuring one metabolite along its line of reasoning, you measure the whole small molecule state of the organism at once, unbiased, then let the data draw the boundary. The label stops being an input and becomes an output. Read off the metabolome rather than assign to it. Every downstream use, hypothesis, mechanism, clinic is that one inversion. This section explains the epistemic inversion that metabolomics brings to aging research. Measure everything first, then let the categories fall out of the numbers. That is the reversal. Classical gerontology runs the other way. It fixes its categories in advance: old versus young, long-lived versus short-lived, fed versus restricted. Then it measures inside a category it has already assumed. The result is self-referential, non-portable, one hypothesis chased at a time. Snapshotting the whole small molecule metabolome reverses the arrow. This means the serum, the cerebrospinal fluid, the plasma, all measured at once. Now, the classifier is discovered from the numbers. The categories emerge instead of being stipulated beforehand, and the dataset stays reprobable when the question later changes. The chapter names 4 problems: classification, hypothesis, mechanism, and clinic. They are one problem. A pre-omic science could only follow a single thread. and it had to presuppose its own distinctions to do so.